Hello everyone, I am Harshita Ghungde from MSc Biotechnology 3rd SEM, and today we will be studying the topic of physical and chemical methods of gene transfer. To start with, we will first understand what gene transfer and artificial gene transfer exactly mean. So, the process in which nucleic acid is transferred from one organism to the another is referred to as a process of gene transfer, and it results in alteration of the genetic makeup of the cell or the organism which receives this foreign nucleic acid segment. The thing here to note is that the organism or the cell into which the foreign gene of interest is being incorporated is called as the host before transformation has taken place, and after the transformation is completed it is referred to as the transformant. Gene transfer is both natural as well as artificial. In nature, gene transfer takes place by two means, vertical as well as horizontal. So, vertical gene transfer is the process in which genes are simply transferred from parents to their offsprings, whereas in horizontal gene transfer, the genes are transferred from one organism to the another, which are neither parents nor they are the offsprings to one another. A classic example for this is the transfer of nucleic acid sequences from one bacterium to the another, which have been infected by the same bacteriophage during bacteriophage infection. Artificial gene transfer, as the name suggests, it is the process in which gene transfer is carried out by humans in vitro conditions. This facilitates us to study various knock-ins and knockouts, to study the function of various nucleic acid sequences in order to support biotechnological researches as well as develop treatments against various diseases such as gene therapies and other treatments in which replacement of genes might be required. To start with, we will first understand physical methods of gene transfer. As the name suggests, here mechanical and physical forces are used to incorporate the gene of interest into the host cell. And the first method here to study is biolistics. Biolistics is also called gene gun or particle bombardment method, and it is one of the most commonly used method to bring about gene transfer and is one of the most successful methods as well. Here, the gene of interest is first precipitated upon the microprojectile particles. The microprojectile particles here are gold and tungsten. So, why we are using gold and tungsten is that there is a characteristic of microprojectile particles which should be maintained, is that they should be biologically inert. What do we mean by this is that whenever we are introducing the microprojectile particle into the host cell, it should not induce any other biological process within the same. So after ensuring this, the gene of interest is incorporated, first precipitated upon the microprojectile particles, and then they are loaded into a machine which is referred to as the gene gun. The target cell is placed at a specific distance from the gene gun, and the high pressure of compressed gas, which is usually helium, is used in order to shoot these microprojectile particles to the targeted cell. When these microprojectile particles are shot, they are responsible for piercing through the cell wall and the cell membrane of the host cell and enter into the cytoplasm. As the DNA enters into the cytoplasm, it gets integrated into the host genome and transformation is brought about, after which we can study the expression of the desired gene of interest within the host or the transformant cell, what do we say it. Advantages and disadvantages of biolistics is that it can be used for a wide variety of cells to bring about gene transfer, and it is an efficient technique. However, since we are talking that the piercing of the cell wall and cell membrane of host cell takes place, it is one of the major limitations that biolistics put forward. Here also in the diagram we can see that the DNA is first coated over the gold particles and then placed into the gene gun. This gene gun is then shot, as a result of which the DNA incorporates into the host cell. These cells are then cultured in vitro conditions to give rise to the plants from these recombinant cells which express the desired gene of interest. The second method is electroporation. Here the host cell is exposed to electrical impulses which in result facilitate the incorporation of gene of interest. What exactly here is happening? Say for example, it is mainly used for plant cells. So the first plant cell is first made devoid of the cell wall, that is the protoplast is extracted. This is followed by placing this target protoplast into a conductive solution. This conductive solution is supplemented with the gene of interest. So यहाँ पे हमारा जो gene of interest है, that is already present within the solution, and then we will use two electrodes. ये usually aluminium electrodes होते हैं, which are used for facilitating the current supply. When the current is passed through the conductive solution, it is passed on in certain patterns. या तो it will be high voltage short time duration frequencies, Alternatively, it can be low voltage, long duration of current which is passed through the conductive solution. This results in temporary disintegration of the plasma membrane integration, and pores are created. So when these pores are created, the DNA or the nascent gene of interest which is present in the conductive solution, it passes through the pores that are formed in the plasma membrane, and incorporation of the gene of interest take place. What happens further is that when these current pulses are stopped, the plasma membrane slowly resettles, that is it reorganizes itself, and by this time the gene of interest had already been incorporated into the host cell, and that transformation has been brought about. Now there are certain factors that can affect electroporation. First, as we discussed, it is the voltage, that either high voltage for short duration is used or low voltage for a long time duration is used. In दोनों conditions में ही what it facilitates is ultimately there is pore formation because the integrity of the plasma membrane is temporarily disturbed. And also the DNA concentration जो हम conductive solution में provide करते हैं, it also facilitates a sufficient concentration of DNA ensures that integration of the DNA into the cell takes place efficiently. This is the diagrammatic explanation here. हम यहाँ पे देख सकते हैं that we have taken the plant cell as the host cell, and again हमने उसका protoplast extract किया है and we have placed it in a conductive solution. This conductive solution is also supplemented with the gene of interest or the target DNA sequence. Now जब हम इसके through current pass कर रहे हैं, so there is formation of pores as I told you earlier in the plasma membrane, and incorporation of the gene of interest has taken place. Again, this cell can be cultured in vitro conditions to give rise to a plant that will be expressing the characteristics that we want in it. Now the next topic to study is microinjection method. Microinjection is one of the most controlled methods to bring about gene transfer. What we do here is first a micromanipulator system is used to hold the target cell. So usually the blunt end of a micropipette is used to create a suction. इस suction की वजह से the cell which is placed over the platform, it remains fixed so that it does not wander upon the entire platform. And then a glass pipette is used. This glass pipette has very narrow tip into which the gene of interest is loaded. This tip is then used to directly pierce through the plasma membrane of the target cell, and then either into the cytoplasm or into the nucleoplasm as per the requirement. We can introduce this naked DNA segment into the host cell. So this facilitates an efficient, however a time-consuming process by which transformation of cells can be brought about. Microinjection is mainly used for animal cells, and it puts forward the advantage of high precision and efficiency. However, it requires a lot of skill and a lot of specialized equipments are required. So microinjection can also be a highly costly process to bring about gene transfer. This is the diagrammatic representation. यहाँ हम देख सकते हैं कि holding pipette is used for holding the target cell in position. The suction is created so that the cell doesn't move much, and then the microinjection is loaded with the DNA of interest and injected directly. Here in the diagram it is directly injecting the gene of interest into the nucleoplasm. Alternatively, as per the requirement, it can also be introduced into the cytoplasm of the cell. Now chemical methods of gene transfer, as the name suggests, यहाँ पे हम हमारे gene of interest को किसी न किसी chemical agent के साथ couple कर रहे हैं in order to facilitate the incorporation into the host of the gene of interest into the host cell. Let us study three major methods, one of which is polyethylene glycol mediated gene transfer. So polyethylene glycol PEG is one of the most widely used chemical methods of gene transfer. Firstly, हमारा gene of interest we have isolated, and we will then mix it with a PEG solution, usually 15 to 20% of PEG. This solution is also supplemented with divalent cations. The calcium ions are also present in this solution. So what we do next is that the host cells are then introduced into this solution. What happens is that this PEG is responsible for temporary disintegration of the plasma membrane, and pores are formed upon the plasma membrane of the host cells as a result of this. Secondly, the calcium ions which are present in the solution, they are responsible to precipitate the DNA segments, the gene of interest. They get precipitated because of these calcium ions onto the surface of the plasma membrane of the host cell. So by both of these methods, by pore formation by PEG, and secondly by endocytosis or phagocytosis of the DNA that precipitates upon the plasma membrane. By both of these processes, the DNA segments enter into the host cell and incorporation of the gene of interest is facilitated. Efficient concentration of DNA is also present in the solution, because जितना DNA concentration उसमें efficiently present होगा उतने ही high chances होंगे that the DNA gets integrated either through the pores or via endocytosis that calcium ions facilitate. Now the second method is calcium phosphate transfection. What calcium phosphate transfection does is that it co-precipitates the DNA upon the targeted cells. Here the gene of interest is first mixed into a buffer solution. It is a phosphate buffer, and secondly a CaCl2 solution is prepared. Now the CaCl2 solution and the phosphate buffer are mixed together, as a result of which calcium phosphate precipitates are formed. What is unique to this process is that the DNA fragments जो हम introduce करते हैं इस solution में, the gene of interest जो हमने introduce किए थे, they get encompassed within these calcium phosphate precipitates, and these precipitates tend to fall upon the surface of the vessel in which the reaction is being carried out. Now the unique feature to this process is that the targeted host cells, they are present at the bottom of the vessel. Like the vessel in which the reaction is being carried out, the host cells are laid at the bottom. So as calcium phosphate precipitates are formed, they fall upon the host cells that are present at the bottom, right? And the DNA is already present within these calcium phosphate precipitates. This is followed by either endocytosis or we can say phagocytosis of the gene of interest. So subsequently the gene of interest gets incorporated into the host cells and transformation takes place. The concentrations of calcium phosphate and DNA are often modified in order to optimize the conditions so that calcium phosphate transfection can be brought about easily, and incubation time and pH are also highly regulated to ensure that all the precipitates are, majority of the precipitates can be taken up by the cell to ensure transformation of the host cells. This is a diagrammatic representation. Here we can see calcium chloride solution is prepared and the gene of interest is present in the phosphate buffer. इन दोनों solutions को हम mix करेंगे and it is not shown here clearly, but in the vessel in which the mixture of these solution is placed, at the bottom of this the host cells are present, right? Then as the calcium phosphate precipitates are formed, as I said on earlier, that DNA is encompassed within them. जब ये cells के ऊपर ये precipitates fall करते हैं, so via endocytosis the precipitates are taken up by the cell and subsequently the gene of interest is also incorporated in the same. The last method to discuss is liposome mediated transfection. What liposomes are is that they are artificially developed lipid vesicles. These are said to have a net positive charge upon their surface, and what this positive charge does is that the negatively charged gene of interest is encompassed within these liposomes, and जब gene of interest हमारा इन liposomes के अंदर encompass हो जाता है, it is referred to as the lipoplex. Now what we do is that these lipoplexes are allowed to integrate with the plasma membrane of the host cells. That is, the host cells are first suspended in a suitable buffer solution and the liposomes carrying the DNA, that is the lipoplexes, are introduced within the same. Because of a net positive charge जो हमारे lipoplex पर present है and the host cells are said to be carrying a net negative charge upon their surface, there occurs an interaction in between the lipoplexes and the target cells, as a result of which the liposome vesicles ultimately fuse with the host cell, and जो हमारा gene of interest है, which was encompassed within the lipid bilayer, it gets incorporated into the cytoplasm of host cell and thereby it brings about the transformation. This is the diagrammatic representation of how lipoplex fusion with target cell takes place. Here we can see that first there is a hollow liposome, that is a phospholipid bilayer. Then we introduce a gene of interest within the liposome. It will be referred to as a lipoplex. Now this lipoplex bears a positive charge and the host cells have a negative charge on their surface. So the lipoplex ultimately it fuses with the host cell, as a result of which, as the diagram shows, the incorporation of gene of interest to the target has taken place. Now there are certain other conditions which must also be considered to facilitate efficient gene transfer. First is the cell type. This is the fact that there are certain cell types which respond best to certain types of gene transfer methods, whereas there are certain other cell types that are suitable for other types of gene transfer. So the thing here is that depending upon which host cell type we are working upon, likewise we select which method of gene transfer we will be utilizing. Say for example, microinjection is most suitable for mammalian cells, liposomes are most suitable for mammalian cells, electroporation bacterial cells का transformation ज़्यादा support करता है, and in the same way biologics हमारे plant cells के transformations के लिए support करता है. So this should be taken into account as one of the earliest and important factors that हमारे होस्ट सेल का टाइप क्या है. ऑन द बेसिस ऑफ दिस वी डिसाइड कि जीन ट्रांसफ़र मेथड एम्प्लॉय कौन सा करना है, बिकॉज इट एनहांसेस द फ्रीक्वेंसी एंड चांसेस ऑफ एफिशिएंट ट्रांसफॉर्मेशन विच हेल्प्स अस फॉर अवॉइडिंग एनी difficulties in bringing about transformations. Second is adaptability. This refers that all the reagent concentrations, incubation, temperatures, pH, etc., all of these external factors are highly optimized such that they can support an efficient gene transfer, so that when we are transforming n number of cells at the same time, we ensure that maximum to maximum transformants are obtained and very less amount of non-transformants are remaining in the gene transfer process. Lastly but not the least, cytotoxicity and safety are an important aspect. When we say physical and chemical methods of gene transfer, physical methods में we are using mechanical forces, and in chemical methods of gene transfer there are some chemical agents by which to which we are adhering our gene of interest, right? So all of these elements have the tendency to cause negative impact upon the host cell. Like if you talk about physical methods of gene transfer, तो उसके अंदर piercing of the host cell हो रहा है, whether we say in biologics or we say in microinjection. electroporation में भी plasma membrane की integration disturb हो रहा है because हम current pass कर रहे हैं cells के through. So these induce a negative impact upon the host cell. It produces a stress upon the host cell. Chemical methods में these chemical treatments do induce a negative impact or stress upon the cells, so it should also be taken in account in order to ensure that what is the probability of getting efficient transformation, but at the same time taking into account that there might be some cellular damages during gene transfer. Lastly, safety is very important. It is the safety of both the handler who's performing the experiment. Secondly, it is about the safety of environment as well as the safety of the cell or organism on which you are working. The handler, firstly, the person conducting the experiments, it should be ensured that the person is safe from any kind of contamination if he or she is working with hazardous bacteria or microorganisms. At the same time, the breakthrough of these microorganisms into the environment should also be avoided. And also, in order to ensure efficient transformation which is contamination free for the host cells, it is important that contamination from the user, from the person who's conducting the experiment into the microorganisms which are being transformed is also prevented. So these are some of the major factors that should also be considered for gene transfer in order to ensure that efficient gene transfer takes place along with safety, which is not only for the organisms that are being transformed but also for the environment and the person who's conducting the experiments. And yeah, thank you for listening to this lecture. I hope you understood the topics. Thank you.